the 27th annual Chappel Memorial Lecture. My name is Gordon Curtin. If you don't know me, I'm Associate Dean of Research and Innovation here at OEC, and I'll be master ceremonies for this presentation. I'll give you a brief background on the Chappel Memorial Lecture and then introduce our speaker. So the Chappel Memorial Lecture was established in 1988 through an endowment by Dr.Peter Chappel, a graduate of OEC, in memory of his father, Herbert James Chappel. And since that inception, internationally renowned researchers have been invited to the University of Guelph to summarize their current work and discuss development and progression of their research programs. The lecture and informal meetings are meant to be a source of inspiration for those engaged in or considering graduate studies in biomedical research. So it's my distinct pleasure today to welcome Dr. Peter Wells as our distinguished lecturer. Dr. Wells is a professor in the Division of Biomolecular Sciences in the Faculty of Pharmacy at the University of Toronto. He received his undergraduate degree in pharmacy in 1971 at Dalhousie University and did a clinical residency from 1973 to 4 at McMaster University Medical Center and did a doctorate in pharmacy in 1977 at the University of Minnesota. He then did 3 years as a research associate at Vanderbilt University. His research interests focus on understanding the mechanisms underlying toxicity of drugs and environmental chemicals that are metabolized to reactive intermediate compounds and the determinants of individual susceptibility to chemical toxicity. His disease areas of focus range broadly from teratogenesis, neurodevelopmental anomalies, and neurodegenerative diseases and cancer development. He's been continuously supported by the Canadian Institutes for Health Research, or the Medical Research Council, as originally called, since 1982. So that's quite the achievement. Congratulations. He's had numerous distinctions, most notably from the Association of Faculties of Pharmacy of Canada, the Pfizer Career Research Award in 2011. In addition to over 100 publications in high-profile, peer-reviewed scientific journals and numerous invited presentations throughout his career, I think the most impressive aspect of his resume are the 5 pages of awards and honors to his graduate students and postdoctoral fellows. I've known Peter for many years. We've sat on the same research grant review panels, we've collaborated on research projects, and we've participated in thesis examinations for our graduate students. Over the years, I've been continuously impressed by the way Peter can effectively design key experiments using the appropriate animal models to solve problems related to important diseases and toxicities. For example, in a 1999 Nature Medicine paper, Peter used just 3 figures to solve the mechanisms of toxicity of thalidomide by showing that it causes fetal malformations in susceptible species, such as rabbits, through oxidative damage that doesn't occur in resistant species, such as mice. Equally impressive is the characteristic clarity of his presentations. So I think we're in for a real treat today.
Thanks for your kind words, Gordon, and I hope it's as impressive as he's alluded to. I'll turn down the lights so you can see the slides. So, Gordon did provide me with some guidance. He mentioned that you would be a mixed audience with very mixed interests, and I'm supposed to motivate students to consider biomedical research as well as inspire the faculty. Time will tell. You can give me your vote at the end. So, with the students in mind, and postdocs, these are the people who should be, of course, getting the award because, as you all know, it's the students and postdocs who do the research and often come up with some of the best ideas. And I've organized them by where many of them have ended up. And you can see this is a bit of a sales pitch for toxicology, but it's a highly marketable training because a lot of people are interested in you. The pharmaceutical industry likes you via a variety of other areas of toxicology that are hard to classify, forensic toxicology, on the organizing administration of health organizations. Some are doing postdoctoral fellowships now. A number of them have gone into academia. These are all in Canada, well, except for one in China. Whoops, one in Portugal, too. Consulting industry, research, venture financing. Having said that, I never got any of it, but they're having good careers in it. Health Canada in the federal government, and Environment Canada in the federal government. Minister of the Environment, Health Administration. So it's a broad range of areas that you can go into when you have interest in toxicology, and I'd encourage those who are thinking of it as a potential career to consider it seriously, because you can do almost any area of basic science, but with a toxicological focus, and it makes you marketable, besides being fun. I'd just like to point out that undergraduate students have contributed to the research. These typically come from gifted programs and work almost as hard as undergraduate university students when they come. In the past, a lot of undergraduate students have worked in our lab over the years, and many of these have ended up with publishable figures and co-authorships on the papers. And so by the time they've hit graduate school, they've already had a publication. So those of you who are undergraduates in the audience, get with it. There's no time like now to immerse yourself. And then a number of collaborators. Jeff Henderson has worked with our lab in the area of neurodegeneration that I won't talk about. Michael Wiley, some of you may know, is an embryologist at the University of Toronto who keeps me from making a fool of myself because my training's in pharmacology and he actually understands embryology. So a brief outline, I'll tell you about toxicology, which is at this university sort of like bringing the coals to Newcastle, because you have one of the preeminent programs in toxicology in Canada. A little about teratogenesis, and then something about reactive oxygen species that I'll refer to as ROS, and oxidative stress and teratogenesis. Talk briefly about the effects of ROS, a little bit about signal transduction, mostly oxidative macromolecular damage, and particularly the example of DNA. And then I'll take you through some actual data. Examples of catalase, the antioxidative enzyme, a couple of DNA repair proteins, and NRF2 that regulates a number of these pathways. Many of you will be familiar with them regulating drug metabolism, but they also regulate DNA repair. So toxicology, most people think of as mechanisms by which drugs cause disease, which is drug toxicity or drug side effects. And one of the things I'll try and do as I go through this talk for the sake of the students is to, sorry, I got to push my timer so I know when to shut up, is to give them ideas and concepts that may not be obvious to people outside of the field. So most people think of it as the study of mechanisms of drug toxicity, which it certainly is. But what I hope to convince you is that it's also very relevant just to the study of disease, which are typically the result of inborn errors of metabolism or cellular processes, pathogens, malnutrition, radiation, etc. And so in some cases, the drug can serve as a probe to understanding a disease. Many of you may be familiar with MPTP, the infamous contaminant of meperidine synthesis that caused Parkinson's disease in young people in the 1980s, and we went from knowing nothing about Parkinson's disease as far as the mechanism to all of a sudden, within 5 years, we knew a lot about Parkinson's disease. Having said that, we still can't cure it, but at least we understand it much better. And associations recently of alcohol with some of the aspects of autism spectrum disorder. In addition, a lot of the models that toxicologists develop turn out to be quite relevant to understanding diseases. And I'm going to show you in this talk some examples, one using catalase and one using a model with OG1, which is a DNA repair enzyme I'll explain later. So in some cases, now there's no drug here at all. It's the model that was developed to examine the role of these proteins in disease and how that can tell you a lot about why things just go wrong normally. So on to teratogenesis. For those of you who are developmentally challenged, there's conception, an embryonic period where you particularly develop structures, organs, that sort of thing. And then a fetal period, the last 2/3 of pregnancy, where particularly you develop functional things like brain function. And then you're born. Now, brain function actually continues on until humans are about 20, but certainly a lot happens during the fetal period. And so xenobiotics can cause teratogenesis, or formation of birth defects. And traditionally, that's been considered morphological birth defects, like shorter absent limbs that you get with thalidomide, or heart defects with phenytoin, and all sorts of other things that you recognize, spina bifida, that sort of thing. There we go. But teratogenesis also refers to any other permanent condition that doesn't go away shortly after birth. And that includes things like cognitive deficits in children who then grow up to retain those deficits that you get with ethanol, or postnatal cancer in children exposed in utero to diethylstilbestrol, or immune system dysfunction that you get with dioxin. And these are permanent imprinting of the embryo into their later life. So that has evolved. So I had teratogenesis up here. This relates to the title of the talk, The Fetal Basis of Adult Disease. And it's a popular thing in the literature, but it only refers, of course, to the fetal period and really doesn't encompass well the embryonic period, which also happens. So then there's a new title. You have to keep changing the title to get funded. And so now we've got, in the literature, you'll see the developmental origins of health and disease. And there is even a society for it now. You know when it's made its mark when there's a society named after it. So it's DOHAD. And this arose primarily because of the growing understanding of the role of environmental stress, independent of xenobiotics, in things going wrong. And this includes, most famously, the metabolic syndrome, was a complex series of problems in adult life relating to diabetes, obesity, and cardiovascular disease that is related. One of the first areas they found was in children during the Second World War, born in Holland, to malnourished mothers because of the German embargo on food. But it also can result from things like psychological stress, pathogen exposure, and that sort of thing. So it broadened it to developmental origins, which of course includes not only the embryonic and fetal period, but also in the early period postnatally. And then it includes things like inborn errors at the molecular level and things like autism spectrum disorders. So that's all I'll say about teratogenesis. And now a little about oxidative stress for those who aren't familiar with it. So oxygen, by a number of endogenous pathways, is converted to reactive oxygen species, and typically that includes hydrogen peroxide, some stuff you dye you bleach your hair with, superoxide, and the infamous hydroxyl radical. And these play essential physiological roles in how your cells signal, how your body develops during embryonic development, and how your cells function as adults. And normally these aren't a problem because you've got a whole host of antioxidant systems to protect you, so it really is kind of irrelevant biologically. However, when you get some drugs and ionizing radiation, you can get an enhanced formation of reactive oxygen species. This can lead to oxidative stress and disease. But I'll also try and show you some examples and remind you that you can also have reactive oxygen species causing disease in the absence of oxidative stress. So even normal levels of reactive oxygen species, if you're not wired properly, can do you in. So biochemical overview with the outcome at the bottom here. We've looked at a lot of teratogens over the time, all of which are based on the idea of oxidative stress, but I'll only show you data for ethanol and methamphetamine today. And in some cases, we're looking at structural birth defects, but mostly neurodevelopmental deficits. And by that I mean when the pups, these are all mouse studies, are born and grow up to adults, they have defects in things like motor coordination, learning and memory, activity, curiosity, and olfaction, other things that reflect subtle brain function. And I'll show you data using a number of models in vivo, which is highly relevant, and embryo culture, which allows you to separate embryonic mechanisms from maternal mechanisms, because oftentimes people think about these things happening in the mother, but in fact reactive oxygen species are too reactive to go from the mother to the embryo. So it all has to happen within the embryo. And the embryo culture studies show clearly that the embryo can form these reactive oxygen species, and it has all the pathways for getting rid of them. And if there's any problem, then the embryo is in trouble. So superoxide is converted to hydrogen peroxide and produces this highly reactive hydroxyl radical. It's sufficiently reactive that it doesn't even leave the area of the cell it's formed in, let alone travel to other cells. And this can damage virtually every macromolecule you've got. DNA and RNA and protein and lipid are some of them, but not all of the molecules. And nobody really knows in a lot of cases what all this damage means in terms of toxicity. There's also a role for signaling with reactive oxygen species, particularly physiologically, but if it gets excessive, that can be a problem, and I won't talk about that today. So xenobiotics can enhance reactive oxygen species formation through a number of ways. They can be converted by enzymes to free radical intermediates. They can produce metabolites that will redox cycle, and they're continually throwing off reactive oxygen species. They can slow the heart, and then when it speeds up again, you get reperfusion production of reactive oxygen species. They can enhance enzymes in the body like NADPH oxidases, so you get a stimulation or an induction of that enzyme, and the enzyme's physiological function is to produce reactive oxygen species. So if you get too much enzyme, you get enhanced ROS formation. And as many of you know, if you interfere with the electron transport chain in the mitochondria, that will throw off reactive oxygen species as well. So there's a lot of ways to generate ROS through xenobiotic exposure, but as many of you will know, there are also endogenous sources that do the same thing. And so you can begin to see that the models developed to study xenobiotics can be very relevant to a lot of diseases that don't involve xenobiotics. And the reasons that we don't all die from all this oxidative stress is that you have a lot of enzymes in your body that will detoxify, particularly hydrogen peroxide, and get rid of everything. Now, even if you get damage, though, to cellular macromolecules, and particularly to DNA, you can repair the damage and still not result in toxicity. And there are a number of different proteins that are involved in this reaction. Now I'd like to just give you a quick aside on different types of ways that xenobiotics cause toxicity and how the difference is really profound. So most drugs, if you've taken any pharmacology, they bind reversibly to a receptor, and if the binding is sufficiently high, the cell doesn't work right and you get toxicity. So there's where you get the dose makes the poison, which is true of most drug toxicity. You take too much, or at least the plasma concentration gets high enough, you get a high concentration of the receptor site, and it's pretty predictable. Don't take too much drug and you won't get sick. It's very different with reactive intermediates. The sequence going from xenobiotic to bioactivation, producing reactive oxygen species, and DNA damage and toxicity, again, this can be mitigated by DNA repair, the ROS themselves, by antioxidants like vitamin E and C, and antioxidant enzymes that detoxify the reactive oxygen species. What you have is a balance between the formation of reactive oxygen species and these mitigating pathways, and there are a few others that I won't talk about. So this balance is what determines the risk. This means that even at a therapeutic concentration of a xenobiotic, or an environmental level, the level of an environmental chemical that someone's exposed to can be a safe level, as defined by the EPA or Health Canada or whoever. But if you've got an individual who either forms too much, in this case in the embryo, which may be different from the mother, converts too much and produces too much reactive oxygen species, or lacks the machinery for mitigating that ROS formation, you can be at risk. So that's completely different from normal things, normal drugs, toxicity, where you have to have at least an elevated plasma concentration, if not an elevated dose. So it's very different in terms of risk. And all of the big epidemiologic studies you'll see do not take these factors into account. And you can have subsets of the population that won't be discovered for years and years and years because this sort of thing is going on and you don't have enough people exposed that are biochemically susceptible to reveal them. So a lot of the things you read in the newspapers, don't believe them. So this is the scheme I just showed you. What I'm going to tell you a little about is signal transduction and particularly oxidative DNA damage. Then I'll take you through some studies on catalase and why, as an example of all of these, this is quite unusual. A little bit about DNA repair, particularly focusing on guanine that I'll define later. And some of you may be familiar with BRCA1 for a totally different reason in breast cancer. And finally end up with NRF2, which is a ROS sensor that will not only enhance a number of pathways involved in protecting against oxidative stress, as well as drug metabolism and some other things, but it also will regulate at least some of the DNA repair proteins that protect you from DNA damage. So first, signal transduction and macromolecular damage. You get ROS. And signal transduction, which is a reversible type of reaction that you need physiologically to survive, versus oxidative damage, which is irreversible. The only way you get rid of that is by either repairing the DNA or deleting the protein. The signal transduction at physiological levels is essential for life, and at higher levels can be pathophysiological and can lead to teratogenesis. In the case of oxidative damage, you can get damage to proteins like carbonyl formation, damage to DNA and RNA, and with respect to DNA, particularly the formation of 8-oxoguanine that I'll talk about later, and damage to lipids. That can be either membrane peroxidation, which is where the whole oxidative stress theory first began to be understood, and the formation of highly reactive products like aldehydes that can covalently bind, and all of these things theoretically can contribute to teratogenesis. So I'm not going to talk about signal transduction, although it likely does play a role in birth defects, particularly at higher drug levels. What I will talk about is oxidative damage. Proteins and lipids are probably targets where you get oxidation that can affect development, but it's hard to prove. And so what I'm going to focus on is DNA as a target, and particularly a specific lesion, one of about 20 lesions that ROS caused, the formation of 8-oxoguanine, because, as you'll see, there are some models that allow you to determine whether this plays a causal, pathogenic role in the outcome versus just being an association or even a downstream event that doesn't really matter. So what I mean by 8-oxoguanine formation is the hydroxyl radicals that are so reactive can react on the 8 position of guanine to produce this 8-oxoguanine. And it's been known for decades that when you get this lesion during cell division, it causes mutations that can lead to cancer, but it also can alter transcription. And that can lead to things like embryopathies, or something I won't talk about today at the other end of life, can lead to neurodegeneration. So many of you will be familiar with the idea of oxidative damage to DNA. You get a mutation on cell division. This is a genetic effect. You've got a change in the DNA sequence. And in that cell, you get a clonal expansion of the cell. And if that happens in utero, then you get postnatally the progeny with a cancer. For example, cancers in children, female children exposed in utero to diethylstilbestrol. However, another consequence is modifications to DNA, histones, and RNA. For example, methylation of genes, acetylation of histones, and epigenetic effects where there's no change in the gene sequence. And this leads to altered gene expression in many cells, which can result in cell death and altered processes within the developing embryo and fetus in this case that can lead to birth defects, either morphological or functional. So there's some question as to how this happens when you end up with modifications to your DNA and histones and RNA that are epigenetic, that don't involve change in DNA sequence. And some of the possibilities are direct oxidation of nucleotides and histones, or indirectly, by altering the enzymes that modulate these things, like DNA methyltransferases and dimethylases, and histone acetylases and deacetylases, although there are a number of other pathways as well. And at this stage, all I can tell you is that this is an area that we're working on. Not a lot is known about it, but I can tell you in the models we've used that DNA damage certainly alters these in a very explicit way. And it looks like there's much to be learned in this area if you happen to be wondering about graduate students, what area you might go into. This is a very interesting area of developmental biology that, with respect particularly to birth defects, is not well understood. So with ROS detoxification, there's a whole raft of enzyme systems involved. We've worked with some of the ones in blue that we've shown clearly play a role in protecting the developing embryo and fetus. And I'm going to show you the example of catalase. So we're up here. Catalase converts hydrogen peroxide to water and oxygen, and hence protects the embryo from oxidative stress. Now, we're particularly interested in the endogenous enzyme, and you might wonder why. Catalase has been studied in all sorts of models by all sorts of people, and it's an important antioxidative enzyme. But in the embryo, it's only 5% of maternal activity. So in classical pharmacology, pathways that are 5% of anything aren't worth studying and certainly won't get you a CIHR grant. So what I hope to convince you is that quantitatively minor enzymes can be very important for ROS-mediated toxicities. So I'm going to show you results from two animals that we've used. These are genetically modified mice that either have low endogenous catalase, acatalasemic or ACAT mouse, or high endogenous catalase activity. So these are mice that express human catalase and have higher levels of the enzyme. And then I'll also show you some complementary confirmatory studies where we've administered exogenous catalase conjugated with polyethylene glycol to stabilize it. And that's you'll see that as PEG catalase. And that's a nice tool because this is a catalytically active enzyme that only has one thing. It detoxifies hydrogen peroxide. If you use chemical probes, they might have a whole bunch of other effects unrelated to that. So first, we're looking at endogenous catalase now, not the stuff you give, but what's in the embryo normally. Less than 5% of activity, and the model we're using is ethanol. It's an important chemical because, as everyone knows, it causes the fetal alcohol syndrome, and that's important because you have to have something like that to get your grant funded. Having said that, there are a lot of other environmental chemicals that would be equally important. You just wouldn't get your grant funded to study them. So the studies I'll be telling you about were done by Latvia Miller-Pinsler. And this is looking at fetal weight as sort of a gross measure, your grams on the y-axis, and these are increasing doses of ethanol on the x-axis. And what you're comparing are wild-type animals. So these are litter mates. These are all in the same litter. One mother, 16 pups. Some are normal activity of catalase, and in this case, some have HCAT, an elevated level, all in the same litter. So the exposures are identical. And so you can see that the wild types lose weight significantly. Weight's tightly regulated, so although these changes are small, they're significant in the wild types. But the animals with extra catalase activity, at least at the 0 dose and the low dose of ethanol, are significantly protected. And if you look at a number of other outcomes, including head length, head diameter, crown rump length, resorptions, and a spectrum of anomalies or birth defects, the wild-type animals are worse off than the animals that have high levels of catalase, which should be protective and indeed do protect the embryo. In this case, we confirm that effect by giving exogenous catalase. And so now you're looking at the effects of giving exogenous catalase. So the wild types in this case are the open circles, and the little triangles enclosed are the ones that have received a pretreatment with exogenous catalase. And in several cases, the weight, head length, crown rump length, and head diameter, there's a significant protection. And actually, I don't have a bar here, but there's also protection with structural birth defects as well. So flipping the coin now, and now you're looking at animals with low catalase activity. So these are mutant mice that have catalase activity, but it's even lower than the 5% you'd normally see. And again, exposed to ethanol, same thing with fetal weight, and it's just flipped. So in this case, the open circles are lacking catalase, and you can see that they are significantly worse off. They lose much more body weight as you increase the dose of ethanol. And that's true for a number of parameters, the crown lump length, which is kind of the length of the fetus, and the head length, and total anomalies or birth defects. And again, all of those effects are rescued by giving exogenous catalase, showing that these mice that are mutant for low catalase aren't different because of some other thing. These are all rescued when you give them back the catalase. Now, more interestingly, getting back to my comment about the models toxicologists developing being useful in the study of diseases, the mice are also interesting to look at from the point of view of untreated animals. And this is looking at physiological ROS production then. And so in untreated animals, if you look in the vehicle treatment, and these are resorptions, which are animals that have died in utero, and they're resorbed. And you can see here that in untreated animals, so again, wild types are in the solid circles, and mice that are catalase deficient are in the open circles. And you can see that the resorption incidence, even in untreated animals, is hugely different. This is 50% of the litter being resorbed, comparing to something that's about 10% in normal. And we've seen this with other antioxidative enzymes like glucose-6-phosphate dehydrogenase. So this is a reproducible phenomenon across several antioxidative proteins, that even physiological production of ROS in the absence of the right antioxidative machinery, and you can get into trouble. Now, in terms of the actual proof of oxidative damage, as opposed to the outcome in terms of the development of the embryo, we can actually measure the formation of 8-oxyguanine with either high-performance liquid chromatography and electrochemical detection, if you want to look at the explicit lesion, or with an ELISA approach, which is quicker and cheaper. And so, again, we're looking at transgenic mice with expressing human catalase. So these are high catalase mice. Again, we're looking at ethanol treatment, only now on the y-axis we're looking at DNA oxidation. And so we've got wild-type mice, either with ethanol or vehicle, and mice with high catalase, either with ethanol or vehicle. This is the endogenous amount of DNA oxidation you get in a normal mouse with only the saline vehicle. And remember, these are all litter mates. And you can see that ethanol is pretty impressive at producing oxidative DNA damage in the wild-type mice. But in the mice that express high catalase, they're completely protected. There is no increase whatsoever. So that 5% of maternal activity of catalase is night and day in protecting the embryo from oxidative DNA damage. And if you give these mice exogenous catalase, so this is pre-treating them with stabilized catalase, you can bring that back to normal. So this level of DNA oxidation from PEG catalase pretreatment is really not significantly different from the vehicle control. So now flipping around and looking at the animals that have low catalase, again, same thing, we're looking at DNA oxidation at the top, only this time we have acatalasemic mice. And you can see ethanol in the wild-type mice, again, produces a substantial increase in DNA oxidation. But if you look at the litter mates that are deficient in catalase, not only are they significantly worse off than the vehicle controls when they're treated with ethanol, but they're also significantly worse off than the wild-type mice. And similarly, when you treat these animals, acatalasemic or catalase-deficient mice, pre-treat them with exogenous PEG catalase, you can totally block that DNA oxidation. So these are effects that are due to ROS formation and not due to some other weird effect in mutant mice. So that's all I want to say about antioxidative enzymes, now DNA repair. I'm going to show you some data in knockout mice for oxyguanine glycosylase 1 or OG1 and breast cancer susceptibility gene 1, or BRCA1. In the case of OG1, I'm going to talk about both untreated and ethanol exposed, because the untreated animals show the importance of physiological levels of reactive oxygen species. And you do not need oxidative stress to be at risk. So we're down here at DNA repair, and first I'll show you data for OG1. Again, this study was done by Litvia Miller-Pintzler. And OG1, or oxoguanine glycosylase 1, is the major enzyme for the repair of oxidative DNA damage, specifically repairing this 8-oxoguanine lesion. And the approach we used was in OG1 knockout mice exposed to ethanol. And here I'm going to be showing you the first results using a test of brain function as opposed to morphological endpoints. So this is a passive avoidance test that reflects postnatal learning and memory, ethanol exposure during the fetal period when there's functional development, and it's the treatment of the mother or the maternal dam in vivo. And I should point out that this is a single injection, one time only, of ethanol in a barely toxic dose. So this is not chronic ethanol exposure. This is a single exposure. But first, untreated. And this is perhaps the biggest surprise and most interesting in some respect. So on the y-axis, we've got the latency to enter a dark chamber, which is how smart you are, basically. And on the x-axis is the treatment. The vehicle wild-type animals, heterozygous OG1 deficient animals, and homozygous OG1 deficient animals. In all cases, vehicle versus ethanol versus pretreatment with PBN, or phenylbutylnitrone, which is a free radical spin trap agent that abolishes the toxicity of a lot of drugs that produce free radicals. So this is the normal performance in a normal mouse in terms of smartness. If you lose one copy of the OG1 gene, you have a significant decrease in your cognition, in learning and memory, reflected by this latency to enter the dark chamber. So the geneticists who argue that you can use a heterozygote as the control, this does not work for a lot of drug toxicities. This heterozygote is significantly different from the wild-type normal litter mate. So these are all pups from the same litter. And again, an even greater decrease in the homozygous knockout, albeit this is not significantly different from the heterozygote. It is different significantly from the wild type. So this is actually the first evidence for an OG1 phenotype, and it's funny because this OG1 mouse knockout was developed by cancer researchers. And because of the obvious relationship to cancer, you'd expect it to be susceptible to cancer. It's not. But it is susceptible to neurodevelopmental deficits. And I'll have a comment about this later on in a minute. And this shows the toxic potential of physiological ROS levels. So these animals have not shown any increase in ROS production. They just can't repair the normal damage you'd get from physiological ROS levels. So there's no oxidative stress here. This is just physiological ROS levels can do you in if you're not properly equipped. Okay, so then what happens when you give ethanol? Won't be a surprise. It makes you even worse off, even in the wild types. It's also a significant decrease in the heterozygotes, although this isn't, the ethanol group is not significantly different from the wild type ethanol group here. And then the homozygous animals, again, a further significant decrease. And there is a gene-dependent effect that the homozygous knockout is significantly different from the wild type. So it clearly is protecting at the level of the latency to enter with ethanol. So this shows the developmental importance of DNA, and also shows you that 8-oxoguanine itself is a developmentally pathogenic lesion. It is not just mutagenic, it is also developmentally pathogenic in terms of, in this case, learning and memory. So then looking at the effects of a free radical spin trapping agent, in all cases across, there is a protection that almost restores it to normal, learning and memory to normal, although not beyond the baseline for that particular genotype. And just a comment about fetal body weight, because it's a general measure of health, and when you give too much ethanol, things typically get worse for the developing embryo. And these are actually very low doses of ethanol for this particular strain. So this is looking at fetal body weight across the wild types, heterozygotes, and knockouts. And the actual message here is there is no difference. These are not doses that are even high enough to alter fetal body weight. And there are also no differences in birth defects. And this just shows the exquisite sensitivity of the developing brain, as opposed to all the other things that ethanol can do to you. And so this is really a threshold dose that we're dealing with. So the conclusions of this sort of DNA repair stuff to date is that adeoxyguanine is an embryopathic lesion, and deficiencies in repair may constitute a risk factor for neurodevelopmental deficits, and another day I could show you the same data for neurodegeneration at the other end of life. And it's similarly protective. The studies in OG1 knockout mice, we found the same thing, protecting against the effects of another ROS inducer, methamphetamine. And we've also found similarly protective DNA repair roles for P53 with a number of drugs, ataxia-telangiectasia mutated with ionizing radiation and phenytoin, and Cockayne syndrome B protein with methamphetamine. So this is quite consistent amongst all the DNA repair pathways we've looked at. It's just that OG1 is the most specific to actually being the hands-on repair protein for adeoxyguanine. So there are a number of comments that are just kind of interesting out of all of this when you think of DNA repair and embryogenesis versus cancer. Most people will be familiar with DNA repair genes as tumor suppressor genes because that's how they were discovered. The cancer guys have all the loot, so they do all the studies, have the money to develop the models. But from an evolutionary perspective, for most of evolution, no one lived long enough to get cancer. You were lucky to make it to your 30th birthday. So aside from a few childhood leukemias that they might have gotten back in the beginning of time, you were dead before you got cancer. So really, all of these genes that the cancer guys study really evolved because of the importance of society reproducing. A little bias there. So some additional observations that are general with DNA repair deficient mice. You can be more susceptible to embryopathies than to cancer. And the example there is what I showed you with the OG1 knockout mice exposed to phenytoin, or ethanol. They exhibit postnatal neurodevelopmental deficits just spontaneously, whereas they don't get cancer. Also, you may be susceptible to embryopathies only when stressed. And where we first saw this was in P53 knockout mice. So the landmark paper that came out in '92 showing the importance of P53 in cancer was from Donhauer's group, which made the mistake of saying developmentally normal, implying that no problem, these guys are just fine throughout development. But in fact, that's only true if they're not stressed. And in fact, when they're exposed in utero to benzoate-pyrene, all of a sudden, these mice show a highly sensitive increase in birth defects. So again, the relevance of most of these cancer genes to development is really important. Other DNA repair genes leads on to the next possibility of BRCA1. And these studies were done by Aaron Shapiro and Lutfia Miller-Pinzler. So most of you will know of BRCA1 because of its importance in 2 to 3% of all breast cancers, with an up to 80% risk of cancer with a single mutation in BRCA1. And that's almost now common to just anybody who reads the newspaper. And what I will try and convince you is that it's not only an anti-cancer gene, but it's developmentally protective. So we're down here at the bottom again with DNA repair, but now we're talking about BRCA1 instead of OG1. And this is an interesting protein because it actually is a caretaker gene, and this protein is involved in co-regulating a number of DNA repair pathways. And Aaron developed a conditional knockout mouse model that became BRCA1 deficient just after implantation. And this is a modest decrease in BRCA1 protein levels. So these are not knockouts. And in fact, these mice would reflect people with BRCA1 deficiencies that very likely would not get cancer. So he looked first, exposed them to ethanol on gestational day 9, which is near the start of embryogenesis. And then we looked at a number of measures of DNA oxidation and DNA damage and morphology. And then, so that's during the embryonic period, dosing them with a single dose. And then in separate studies, dosing them on day 17 during the fetal period with a single dose again. And in that case, looking at a test of learning and memory, the passive avoidance test. So we were looking first at the DNA damage, specifically the 8-oxoguanine lesion. And what you're looking at are the open circles are the wild type animals, and the deficient, the black circles are the BRCA1 heterozygous deficient. So these are only heterozygous deficient. They're not homozygous deficient. Exposed either to saline or ethanol. And it's a little complicated, but I'll take you through it. So saline versus ethanol, you see a drug effect. And this is looking at the wild type exposed to ethanol versus the deficient, and there's a significant increase in the DNA oxidation in the deficient pups versus the litter mates that have normal BRCA1. So there is clearly a protection by BRCA1 against 8-oxoguanine in the repair of that lesion. You can see the same type of outcome looking at gamma H2AX, which is recruited to double-strand breaks when you get oxidative DNA damage. And again, the open circles are wild type. The closed bars are the deficient mice, litter mates. And again, you see a drug effect with ethanol, in ethanol versus saline, but you also see a gene effect where there's a doubling in the amount of DNA damage in the deficient progeny that are exposed to ethanol versus the BRCA1 normal litter mates exposed in the same litter. So finally with BRCA1 then, looking postnatally, this is the outcome now in terms of the actual morphological consequences. So this is looking in embryo culture during the embryonic period, exposure during the embryonic period when organs are formed. So what we're looking at here is saline and comparing wild types to knockouts with a number of parameters. I won't go through them, but you can evaluate embryos in a number of morphological ways to determine if they're developing normally. And being knocked out, these are the conditional knockouts, remember, heterozygous, all these bars are the same. So they develop normally. So BRCA1, a heterozygous deficiency, really doesn't affect development. If you give the wild type animals ethanol, at the concentration we're using here, again, all the bars are the same. So this is ethanol versus saline for, in this example, somite development. And for all of these, there's no effect. So this is a dose of, a concentration of ethanol that does not alter embryonic development in this strain. On the other hand, if you look at the knockouts now, it's a different story. Here you see that the knockout exposed to ethanol is more affected significantly than the knockout than the wild type exposed to ethanol. So the loss of one gene makes you more susceptible to change in somite development, and there are similar changes in a number of other parameters. So this clearly is also, in addition to protecting you against DNA oxidation, it's also protecting you against embryopathies. And then finally, we looked in vivo in some preliminary studies where we exposed them once to a single dose of ethanol during the fetal period by treating the mother. And again, you see a gene effect. In the ethanol-exposed progeny, the deficient animals have significantly lower learning and memory abilities than their wild-type litter mates who were exposed in utero to that single dose of ethanol. So it appears that BRCA1 is indeed not only an anti-cancer gene, but is developmentally protective. And we've got other evidence that similar protection against other ROS-inducing drugs like methamphetamine. So this probably has a fairly important role in protecting the embryo against oxidative stress from other factors. So then the final thing is NRF2, which some of you will know is nuclear factor erythroid 2 related factor 2, hence why nobody calls it that. And the question is the developmental role. So a lot of people are familiar with NRF2, and I'll show you why in a minute, but the developmental role is much less clear. And the hypothesis was, does this provide fetal protection against methamphetamine, which is a ROS-inducing teratogen? And these studies were done by Anne-Marie Ramkissoon. So when you get reactive oxygen species, it activates NRF2. NRF2 is known broadly as a ROS sensor. And it has a whole bunch of roles in terms of upregulating genes relevant to oxidative stress, heme oxygenase, drug metabolizing enzymes, particularly phase 2, a number of antioxidative enzymes, and at least one DNA repair enzyme, OG1, and very likely others as well. So we're here then with NRF2 as a ROS sensor upregulating antioxidative enzymes and at least one critical DNA repair enzyme. And previous studies had looked at ethanol and a model where you produce reactive oxygen species measured by fluorescence and DNA oxidation measured by 8-oxyguanine, and not embryopathies, but at least apoptosis, which many people believe will lead to embryopathies, and at least in some cases certainly do. So ethanol will initiate that chain of events. That was by Kathy Sulik and some collaborators. And then they used a NRF inducer, D3T, and they showed that if you gave that prior to ethanol, you had increased NRF mRNA, protein binding, and binding to the ARE site on the gene, which resulted in an upregulation of a number of NRF target genes for antioxidative enzymes. And this, when you pre-treated the animals with this inducer, blocked the production of ROS and blocked the effects on apoptosis, suggesting that it very likely was going to be embryo protective. And there was a functional NRF pathway, at least in their embryo culture models. So we looked at this with fetal neurodevelopment in vivo using NRF2 knockout mice because we were worried that the probe, DT3, would have other effects and it would be a cleaner study to use a knockout. And we gave them methamphetamine in vivo during the fetal period to look at their postnatal neurodevelopment. So I won't go through all the production of this other than we gave the drug, and then on day 17, I'm actually going to make it. Looked at a number of outcomes in terms of NRF activation, DNA oxidation, and toxicity. And crucially, I'll show you the data for activity and then allude to some other results to show you what the outcome was. So what we're looking at here is NRF2 modulated gene transcription in the fetal brain, comparing the NRF knockouts, specifically in this case only the wild types, to the homozygous knockouts. And we looked at a number of readouts. Heme oxygenase is one, and ADPH quinone oxyreductase 1, which are well known to be modulated by NRF2 in adults. And because we're interested in this one particularly, oxyguanine glycosylase 1, or OG1. And what we found, it's going to be hard for you to see these, but basically these are the wild types with normal NRF2, and you can see they show this induction of heme oxygenase 1 mRNA, whereas the knockout completely blocks it. So this shows that in the fetal brain at least, there is an intact NRF2 pathway, and heme oxygenase is absolutely dependent on that NRF2 pathway. Same for NQO1, absolutely dependent on NRF, and the same for OG1, absolutely dependent on NERF. So the developing fetal brain, at least, is exactly like the adult, only in addition to some of the pathways that are known, it also regulates at least oxyguanine, glycosylase 1, and possibly other DNA repair proteins. So looking at DNA oxidation, you've got DNA oxidation here on the y-axis, saline with the 3 NERF genotypes, and meth with the 3 genotypes. So first, the animals exposed to meth, either the wild types or the heterozygous knockouts, were significantly greater than the saline, showing a meth effect, and the homozygous knockouts were significantly greater than either the heterozygotes or the wild type, showing a gene effect, NERF2 dependent increase in DNA oxidation, which could be, of course, not only the effects on NERF regulating 8-oxyguanine directly, but also upregulating antioxidative enzymes. Now looking at fetal weight loss, the females, again, saline with all 3 NERF genotypes, and meth with all 3 genotypes of NERF. This dose of meth had no effect on the wild-type mice, but there were significant effects in the heterozygotes, and even more significant effects in the homozygous mice, showing a NERF2 gene-dependent effect. And the gene dose response is shown here with the homozygous mice progeny showing more fetal weight loss than their heterozygous litter mates. In males, again, the wild types were unaffected by this dose of meth, again, trying to get a threshold dose that is minimally toxic. Both the heterozygous and homozygous knockouts showed increased weight loss, although there wasn't that same gene dose effect that we saw here with the homozygotes were not significantly greater than the heterozygotes, although the females were. And it's interesting, a number of the behavioral studies we've done with a number of ROS-initiating teratogens have all found that if there is a gender difference, the females are more susceptible. That seems to be a pattern we're seeing. And so finally, looking at an activity monitor to see the effects of NERF. These are, I won't show you the movie, I didn't even attach the clip, but you can look at their activity and actually quantify it. So this is looking at females with distance as a postnatal activity monitoring as effects of activity could reflect both cognition to some extent as well as motor coordination. So you can see a drug effect here. When you give meth on the right to the homozygous knockouts, the effect is significant. There's a sort of a trend here for the wild types, but it's not significant. So it's a drug effect. And there's the suggestion of a gene effect, it's less than p.1, but it's still greater than 0.5, so it's just a trend. Now, in this case, it's the males that show the interesting effect. They show the same drug effect the females did, but here, the males actually do show a gene effect, with the homozygous NERF2 knockouts being more affected by meth than the homozygous normal animals. And there was a similar gene effect when we looked at another complex test that is too tedious to describe, olfactory discrimination, which uses completely different neurons from activity. So this is probably broadly based in the fetal brain, these effects of oxidative stress and their regulation and protection by NERF. But being pharmacologists, we're not mapping the entire brain. We're looking for a proof of principle to determine mechanisms. So NERF2 is developmentally protective, at least for the ROS-initiating teratogen methamphetamine. So in overall summary, I've taken you through signal transduction to a lesser extent, mostly showing you the role potentially of DNA damage in teratogenesis and the role of catalase being important. Even though it's only 5% of maternal activity, it still plays a crucial role, and quantitatively minor enzymes, when you've got reactive intermediates, can have major biological significance. And the role of DNA repair and particularly the broader role of BRCA1 beyond breast cancer in protecting the developing embryo and fetus from oxidative stress. And finally, NRF2, like in the adult, is profoundly important in protecting the developing embryo and fetus from oxidative stress initiated by a number of xenobiotics. And it remains me to thank the Chappel Memorial Lecture Fund for their support and the Ontario Veterinary College for inviting me here. Thank you very much.
Well, I don't think he's disappointed. Thank you very much. Before I open it up for questions, I just want to mention that there is a reception afterwards, just across the hall in the cafeteria. Are there any questions?
Thank you for that talk. I've seen one of your shares of talks, and that's the best talk I've ever seen in the entire.
Wow. I want to get a tape of that for my graduate students. They won't believe it.
It has ramifications that basically challenge what we believe about any person in society. It really does. And we probably won't recognize it before that. But the question is, everything we're affected pushes us towards or away from inflammatory. Have you done any studies that relates to inflammation, systemic inflammation, with respect to all the parameters?
No, but you can guess. The only thing I'd say is it won't be maternal inflammation that matters. And that's one of the big problems is how do you study this in humans? You know, you can't sample them out of it because all of these things are proximate. The reactive oxygen species has to be produced within the embryo. And so all of the studies that suggest safety are based on, you know, it's like looking for the treasure in the shallow water, whereas unfortunately the ship went down in the deep water. Yeah. Well, you can do these sorts of things. You could test them in monkeys, but you'd need more money than the CIHR is going to give you, if it gives you anything at all. So that's the biggest problem is a lot of these things are known to produce oxidative stress. Well, I mean, one way of inflammation is getting exposed to a pathogen that will set up a local oxidative state. But no matter what you do, you've got to study it in the embryo. And you could investigate your hypothesis by doing something that would stimulate inflammation locally. I mean, you can study that in embryo culture too, a quick proof of principle, and then you could use, one of my students developed a neat trick for injecting intraamniotically and then intraventricularly into the ventricle of the fetal brain. So the models are there to do that sort of thing. So if graduate students are interested in inflammation and development, it would be a good thing to do because nobody knows, but you'd predict. It would be hard to believe that it wouldn't be important.
Just at a functional level, do you think that the 8-oxyguanine that you're finding, or that's presumably going up in your OG1 mutants and in some of your other mutants, is it impacting the number of neurons that they have or the functions of the neurons that are there? So if you look downstream, do you find a greater number of neurons in the controls versus your treated animals, or is it a case of you've completely changed the pattern of gene expression from the loci that are oxoguanated, or whatever you call it?
So being a pharmacologist and not a neurologist or neuroscientist, I would keep my options open. And you may recall I had a little box that showed everything from cell division, differentiation, migration, communication. I think it could affect the numbers. I could affect where the neurons go, how they talk to each other. All of those things are possible. And great things for graduate students starting their career to investigate.
Have you had a chance to look at downstream gene expression at all in those samples? We talked about it a little bit when we met, but I don't remember what you said.
That's what we're planning to do, and we're hooking up with guys who spend their life doing whole genome expression studies and chromatin immunoprecipitation with RNA sequencing. And if we ever get the money from CIHR, we've got the perfect model because we can look at knockouts compared to their wild types, and we'll know which changes matter instead of thousands of gene changes you know you're going to see going up or down, most of which are epiphenomenon, we'll actually know which ones are pathogenic. Invite me back in 3 years.
So I was really curious about your data with the catalase experiments where you showed the enzymatic oxidants. I was curious to know if you have also some data with respect to non-enzymatic antioxidants like taurine or ascorbic acid, which might have a similar role, a protective role.
Yeah, we've looked at antioxidants like vitamin E. I mean, in some respects, you could call phenylbutylnitrone an antioxidant. It's a free radical spin trapping agent. We've looked at caffeic acid as a water-soluble antioxidant. They're all protective against reactive oxygen species-mediated teratogenesis. Although phenylbutylnitrone is more effective than vitamin E or caffeic acid. And we don't know whether that's because we didn't get the optimal drug concentrations or what, but they all work to some extent. But antioxidants like vitamin C are the least effective of all the things we've tried.
What's the story with the hypotaurine or taurine in general? Have you looked at those as well? Or?
Haven't looked at those. In fact, when we're looking mechanistically, because of limited budgets and manpower, we try and get the most definitive thing we can. That's why we use catalase, exogenous PEG catalase, because all of those other things, like vitamin E, there are over 30 different things vitamin E does in addition to being an antioxidant. Whereas catalase, it probably has some other role, but it has yet to be identified. The only thing anybody knows it does is metabolize hydrogen peroxide. So we pick the probes as definitively as possible, and since you can maintain the catalytic activity of catalase by conjugating with polyethylene glycol, that's what we use as our antioxidant rather than going through some popular antioxidants.
Good lead into my question.
Uh-oh.
Regarding catalase, it really is remarkable it has such an effect. But I mean, a lot of people think that it's really not that important for you to do just damaging in vivo. But so many backups for peroxidase and glutathione peroxidase. Can you speculate further as to why you see this having such a
No, it could be due to localization things, the relative levels of some of those other redundant enzymes at the same gestational time. Remember, these are in the developing embryo, not an adult. But it's an interesting philosophical question because I didn't show you data for another antioxidative enzyme. Well, I call it one. It's actually an indirect antioxidative enzyme, glucose-6-phosphate dehydrogenase, which produces the NADPH that's used for a whole bunch of other things, including ultimately the detoxification of hydrogen peroxide. And when we started these studies, I remember we had a biochemist on the advisory committee of the student doing it, who in the first committee meeting crushed the graduate student by telling them that this was a guy who'd written a textbook in biochemistry, that he was wasting his time because there were so many pathways for producing NADPH that it just wasn't possible that he would see any effect. Now, some of you may know NADPH was known to have an effect in biology, and people who have a deficiency in it do get hemolysis. So we knew it had some biological link, at least in a cell that can't make, it doesn't have a nucleus, so it can't make anything to react to oxidative stress. I guess another lesson philosophically for the graduate students is don't believe what old people tell you, because until you actually do the study, biology is sufficiently complex that you won't know. So go ahead and do the damn thing anyway, as long as you can get the money from your supervisor. And to make a long to make a long story short, G6PD was very important. Although unlike catalase, it's the only antioxidant enzyme in the embryo that is of the same or higher level than the adult. Whereas catalase is only 5%. But I guess getting back to your point, there are a lot of different pathways for producing NADPH, and that one enzyme, if you don't have it, you're in big trouble. So it's consistent that although technically, we have lots of redundancies to protect us from oxidative stress. That doesn't mean those redundancies will save us. And the reasons for those, will require a lot of people and a lot of money to figure out. And typically they'll have to do it without telling anyone how they're going to spend the money, because some old person will tell them, no, that won't work. Yeah?
When you teach students about oxidative stress and organic compounds, because it will show hydroquinones, quinones, enones, things that can redox cycle, and we won't show them ethanol in that list. So how does ethanol induce reactive oxygen species?
Well, one possibility was that during its metabolism by P450, you get the same sort of thing you see in mitochondria, only a different type of cycle that will leak superoxide. And Dr. Josephi can elaborate that in painful detail. Never really understood how the electrons move myself, but actually what's turned out to be probably more important, and probably is maybe important for a lot of teratogens that haven't been investigated yet, is that ethanol will induce NADPH oxidase, of which there are over 4 or 5 isozymes scattered throughout not only the body, but within different components of the cell, including the internuclear membrane, where it makes superoxide. And you can show in NOx, with NOx inhibitors, that you can block the effects of ethanol, at least in terms of apoptosis. They haven't gone on to look at birth defects or anything. And we've shown with methanol that you can actually block birth defects with NOx inhibitors. So I think that one source for a lot of xenobiotics may well be the induction of NOx, or NADPH oxidase. It used to be known mostly for its ability to produce in macrophages superoxide that the macrophage uses to kill bacteria that's engulfed. But it's ubiquitously expressed in different cell types and throughout the cell in different locations depending on the isoform. And it's a major potential source of ROS, possibly more important than mitochondrial sources that are so popular now when people talk about endogenous ROS production. I think there's a lot of evidence that NOxes are a huge source of intracellular ROS production and possibly intranuclear ROS production. So that would be my guess for ethanol right now, although that study hasn't been done definitively yet. Well, the animal models we used were for methamphetamine and using ethanol. So all of the things that we study in mice, most of those have a correlate in humans. But nobody has looked in humans to see whether any people with the fetal alcohol syndrome, and particularly the behavioral abnormalities, have different levels of catalase or not. And you certainly could at least discover people who are at risk because they were genetically deficient, but that wouldn't tell you if they had been environmentally exposed to something during pregnancy that had reduced their catalase activity. And it probably wouldn't tell you, unless they did the study very unusually, whether there were epigenetic changes, not genetic changes. And so it's quite possible if somebody did the study and didn't do it rigorously, they would actually hide the effect. Because you don't care whether it's an effect on the protein or the gene or an epigenetic effect. All you would like to know is did they have decreased catalase activity in the embryo, not the mother, at the time of exposure. Very hard to do.
Okay, if there aren't any further questions, please join me in thanking.
That was fun. Good questions.
Yeah, they are.